Good evening. Welcome to the 2020 Salvesen lecture. My name is Peter Matheson and I'm the principal of the University of Edinburgh, and it is my very great pleasure to be welcoming you to this evening's event. Thank you very much for joining us wherever you are. This will be the second Salvesen annual lecture, and in itself will be a great learning opportunity, but it also gives us an opportunity to celebrate another year of successful collaboration between the University of Edinburgh and the Salvesen Mindroom Centre charity. I'd like to thank Astle Salvesen in particular for his fantastic ongoing support of this work. This is an area in which the University of Edinburgh is proud of its strengths, but there does still remain so much more to be done. Tonight's lecture will be delivered by Dr. Duncan Astle, who's the programme leader at the Medical Research Council Cognition and Brain Sciences Unit at the University of Cambridge, and it'll be in the form of a pre-recorded film to be followed by a live question and answer session. For this year's event, Duncan has created a wonderful film about the study at the Centre for Attention, Learning and Memory, which has the acronym CALM, featuring the voices of some of the children who've contributed to this project. Duncan has carried out some seminal work with children who find learning difficult, and he was selected as a speaker by the charity Salvesen Mindroom Centre, who had read some of his work and found that it was very relevant to them as practitioners. One of the core principles of this research and the work of the charity is involvement of children and young people with learning difficulties and their families. So here to tell us more is Duncan, together with the most important people that he works with, the children themselves. Thank you for joining us, and now over to Duncan.
Hello everyone, and greetings from my kitchen table. I'm Duncan Astle, and it's my pleasure to give the 2020 Salvesen lecture. I'd like to thank the two Salvesen Mindroom Centres, both the charity and the research centre, for inviting me to give the lecture. It's really humbling for our research to be recognised by such an international centre of excellence, and one that seeks to empower children and young people by placing their voices at the heart of research on learning difficulties. I wish that I could be with you in Edinburgh to celebrate the great work that you've been doing all year. But in my absence, the Salvesen Mindroom Centres suggested that we make this film to showcase our research. And a massive thank you to Cambridge Filmworks, who helped us produce it. In the film, you will hear from families that make our research possible. You'll hear about the research in their words. You'll hear about their experiences. You'll hear how we collect data and how we analyze some of it. So whether you're a practitioner, a scientist, neurodiverse, a family member, or just really interested, I really hope that you'll find something in the film that you really enjoy. Thank you.
School always used to be, I remember being very hard. I used to get told off a lot.
I don't really like school because there's a lot of writing, reading and stuff like that, but like some bits I like about it and some bits I do not like about it.
In secondary school, it's like every lesson you need like 55 minutes. You need to move to a different class. That's what I found a bit tricky.
I used to get up in the mornings when I was younger. I used to always just think, no, I don't want to go to school.
You were quite nervous when she was little. Sort of new situations would sort of fling you into distress.
I didn't really like loud noises, scare noises like that. So like hair dryers, hand dryers, stuff like that, it just makes me really stressed. And then like in year 2, I got kind of used to it and I could go with it.
My reading's like not gone really well, but then my writing has improved like a lot since I was in primary school.
If like the teacher gives me new things, I think it's quite hard, but if I like go over them, like if I'm not at school, if I do it like as homework, I think I'll get it over like maybe like 2 days, 1 day.
Freddie is different to what the textbook case of ADHD.
I don't know what to do. Oh, nothing.
I don't think that the way we study children with neurodevelopmental conditions actually often really reflects the experiences of the children that have those conditions.
He still kind of treated me, harder, more than the other students.
It can be quite tricky to know where to go, what to do, if your children are maybe socialising differently. It's quite overwhelming, it can be quite isolating.
There are some problems with the way that we traditionally study neurodevelopmental conditions like ADHD.
Experts, teachers, all professionals involved with children need to recognise that a child with ADHD isn't necessarily going to present the same as another child with ADHD or a similar issue.
That's often because we rely solely on a type of study called a case control design. So a case control design is one where we define a small group of individuals, usually who have a particular diagnostic label, and we compare them to so-called controls, children without that particular diagnostic label. And that's been the most dominant model for studying developmental disorders for the last 20 or 30 years. But we think that there are some problems with it. The first one is that it assumes that everybody with the diagnostic label, i.e. all the cases, are the same as each other. And in reality, we know that there's lots of diversity and variability, even amongst individuals who have the same kind of labels. So grouping them together might not make much sense. Secondly, we're not able to compare children who have different types of diagnostic label to each other and to children with no diagnostic label. And that's because traditionally, you would never have multiple different case groups in the same design. So you'd never be able to find out whether there are any commonalities, for example, a common cause, that might span across multiple different diagnoses. And thirdly, those studies are often very small. So when other scientific groups try to replicate them, often it can be quite hard to find the same thing. In short, these case-controlled designs assume that children with the same labels are very similar, which means that we lose the kind of diversity that's really present. It means that we can't tease out what might be common or shared across supposedly different categories. And finally, we're finding out that those studies often are not tremendously robust, and that's because they're not done at the kind of scale that's needed to produce robust findings.
So we first became involved with CALM when Eva's amazing speech therapist, Alice, recommended her for the project.
When Freddie was 5 and we'd first discovered that there might be an issue at school, his first teacher sort of raised the issue, and I think it was probably during year 1 that we started having sort of appointments at the ADHD clinic, and the doctor suggested that there was this study happening that we might be interested in being involved with, and Freddie was keen.
In 2014, here at the Cognition and Brain Sciences unit in Cambridge, we started a study called the Centre for Attention, Learning and Memory, or CALM for short. And it would be a different type of study of children who have neurodevelopmental conditions or who are struggling at school. Rather than recruiting children according to strict diagnostic criteria, instead, we would invite specialists working in children's educational or clinical services to refer children to us that they thought were struggling in the areas of attention, learning, or memory. So intentionally generic. We didn't know if anyone would be referred to us, but pretty quickly we soon realized that we had very many families being referred to our study. And by the end, we'd seen almost 1,000 children. Many of the children would have a single diagnosis, multiple diagnoses, or in most cases, no diagnosis at all.
I think it was through the school. They contacted me and asked if I would like Billy to be into some research. They didn't really explain much about it until I got contact from yourselves. And I just thought it'd be a good idea to help other people.
I had a number of questionnaires and surveys to fill in, which was actually quite interesting.
When the families visited us, the children would undertake multiple different cognitive assessments, including all sorts of aspects of cognition that we thought might be important for learning. So executive functions, things like planning, attention, language skills, phonological processing or listening skills, short-term, long-term and working memory. And whilst we were doing that, the parent would complete multiple different questionnaires, capturing data on all sorts of aspects of home life, their child's behaviour, their child's language, their child's mental health. So by the end of the visit, we had built an incredibly rich data set about that child. And as we saw more and more children, we gradually built this cohort of children recruited with a broad criteria and providing very rich and detailed phenotyping of each individual child.
So there was one with a computer in front of me, and I was memorising the dots that were in the corners of the screen. So if I pressed one, I had to memorise where it went last time.
So for example, you said a word and then another word, and you have to find if they're the same start with the same letters or end with the same letters.
On site here at the MRC Cognition and Brain Sciences Unit, we have multiple different types of brain scanner. One of them is called a magnetic resonance imaging scanner, or MRI for short. And all of the children who took part in the study were invited to go through the MRI scanner. They'd first get their chance to practice in the mock scanner, and then they'd have the real scan. And during the scan, we would acquire lots of different types of image about their developing brain. And at the end of the study, the children were invited to provide us with a DNA sample by spitting in a pot, which they were only too happy to provide.
You're trying to understand how maybe somebody with ADHD's brain is different to someone without ADHD's brain, to compare.
When I had my MRI scan, it was like when I walked in, it was a really big machine, and I went on it and it makes a few noises, but it takes a scan of your brain.
The end result is a one-of-a-kind resource, rich data on cognition, learning, behavior, and mental health. Genetic data, neuroimaging, all on a group of 1,000 children that reflect the kinds of children that professionals see day in, day out, rather than the more rarefied or selective groups of children that we typically study in the academic literature. And this resource is already being used to answer very many questions about neurodevelopment. There are already over 60 projects that are ongoing using the CALM data. And I'm going to tell you about just one of them. And I like this project because it gave us the opportunity to try some new methods and also to explore the data, making as few assumptions as possible, and to allow the data to tell us about the ways in which children could be similar or different and how their cognitive skills might relate to their brain development. The project that I'm going to tell you about uses something called machine learning. And machine learning sounds very fancy, but really it's very simple. So every time you search on the internet, you type information into a search window. And what you don't realize is behind the scenes is a machine learning algorithm. And you're feeding it data when you type something in. And what the machine learning algorithm is doing is building a model of you and your preferences. And what you might notice is that at some other time, you see adverts that are really well targeted to you and the things you might like to buy. And that's because the machine learning algorithm has built a really accurate model of you and your preferences. So we used something similar, but rather than feeding it internet searches, we fed it some of the rich cognitive data that had been collected as part of the CALM cohort. We used a particular type of machine learning called an artificial neural network. And to explain how it works, I'm going to use some fruit. So let's imagine we have lemons, melons, apples, and so on, and we can describe each piece of fruit with 3 values, say maybe the size, the juiciness, the acidity levels. Then we take a very simple network. To start with, it's just got 3 separate nodes, and each of those nodes is defined by a set of weights, so 3 weights, and those weights correspond to the same values that describe each piece of fruit. And to start with, we take each piece of fruit in turn, so we start with the lemon, and we decide which node in the network has weights that are most similar to the values for this piece of fruit. And we then allocate that piece of fruit to that node. We then update the weights for that node so they become a little bit more like those for the piece of fruit. And we take the next piece of fruit, we find its best matching node, we update the weights, the next piece of fruit, and so on. So gradually, over time, what it's learning is how pieces of fruit group together because they have very similar properties. So you can see, for example, it's learned that there are melons it's learned that there are berries, and it's learned that there are citrus fruits. But what you can also see is that at the minute it's not really able to distinguish the citrus fruits from the apples and pears. And so a neat feature of this network is that on the next run through, what it will do is create an extra node so that next time it's able to learn that distinction. And a really nice feature of this network is that it can represent hierarchical information. For example, it might learn that there are multiple different examples of berries, and there's a higher order class of fruit called berries. So you might be thinking, why on earth did they bother with this artificial neural network in the CALM cohort? And there are a few really good reasons. So the first one is we don't know much about this novel cohort. And so our idea was that the neural network might learn something about the different children that we'd seen. A second thing is that it can learn about hierarchical relationships. So it can learn about whether there are particular classes of cognitive profile and then more refined versions of it. And thirdly, as we'll see later on, it creates a really nice model of the data. And having a model is really powerful because it means you can use it to make predictions about other types of data. So we fed the data into the artificial neural network. But of course, what we're using isn't information about fruits, it's information about the children in CALM. And we have all that rich cognitive data, so we know about their attention skills, their language skills, their memory skills, and so on. So the question is, what will the network learn? Having fed all the data in, maybe it will learn a structure that we are familiar with, that there are children with autism, children with dyslexia, children with ADHD, or maybe it will learn a structure that's totally novel that we hadn't predicted. So what you can see on this picture are some of the data. So the big blue nodes correspond to the top layer. That's the most coarse representation of the data set that the artificial neural network learnt. Then the next layer down are the pale yellows, and the bottom layer are the reds. So if you look at the top blue nodes, you can see that they provide the kind of landmarks for the cognitive space that the network's learnt. So over on the left-hand side, you find children who have got very high cognitive abilities. And as you move towards the right, you find children who have got age-appropriate skills, maybe slightly better at spatial tasks or slightly better at verbal tasks. And then as you move towards the right, you find pockets of children who have more difficulties in spatial elements of cognition, like spatial working memory and fluid reasoning. And you find children who have got more problems in verbal aspects of cognition, like phonological skills or language skills. And then right at the far right-hand side, you find pockets of children who struggle on all of the different tasks that we gave them. So what the network has learned is a kind of cognitive space that can be used to characterize the sample that we collected in CALM. So the first question you might ask is, does the network just learn about a child's diagnosis? And of course, many of the children had a diagnosis in CALM, so we can test that. So if you imagine that we were to find the locations of all the children who had an ADHD diagnosis, if ADHD describes the kind of cognitive profile a child should have, then all of those children should be grouped together. And what you can hopefully see is that they're pretty randomly scattered across the network. And indeed, statistically, that's exactly what you'd expect by chance. And the same is true for children with autism diagnoses, children seeing the speech and language therapist, and those with dyslexia. In short, the kind of diagnostic label that the child already has when they came to CALM didn't describe the kind of cognitive profile that we observed. And that's something that we've written about a lot, and we've produced some materials for teachers that were published in the Times Education Supplement, which is a really good way of reading more about the idea that a child's diagnostic label might not correspond very well to the kind of cognitive profile they have. So what does this network predict? Well, one thing we tested is whether it predicts the learning measures that we collected in CALM, the literacy and numeracy measures. And we tested that using something called hold-out cross-validation, which I'm going to explain now. So if you imagine we took just 80% of our sample and we trained up our neural network, so we create our neural net of the cognitive space that characterizes the children. Then we take the remaining 20% of the children. We take the first child and we find their best matching unit within the network. So their data weren't used to train the network, but we can find where they would sit if they had been used to train it. We then take the learning measures from the children who actually created that node from the original 80% of the cohort, and we use their learning measures to make a prediction about the learning scores of this particular child. And we see how close we are to that child's actual literacy and numeracy scores. By comparison, we can just take another random node in the network and use that to make a prediction. And that's presumably what we would have expected by chance. We can then take the next child that's been left out, find their best matching unit within the network, take the children from the 80% that were used to create that node, and use their literacy and numeracy scores to make a prediction about this held out child's literacy and numeracy scores. Then we can calculate how close we are relative to if we'd have just chosen a node at chance. And we repeat that process over and over until every child has appeared in the holdout 20%. And at the end, what we've done is build a distribution of how accurate our predictions are based on this network. And what you find is that you're significantly better than chance. So it's a Cohen's d of around 0.5, which is a medium effect size. In short, what you can do with this network is use it to make fairly accurate predictions about a child's literacy and numeracy scores. So, just to summarize that first section, we fed the rich cognitive data from CALM into a type of machine learning called an artificial neural network. And that learnt about the cognitive space that describes the children we had seen. Where a child sits in that cognitive space does not correspond well to any kind of diagnosis that they had. But you can use that model to make a pretty good prediction of their literacy and numeracy scores.
The difference from him coming here the first time and the second time, seeing the progress that he's made, is massive. Because we were told that no one knew much about it and we just had to kind of go with the flow. We didn't know where he'd end up or what he'd be.
It's been a very positive experience being involved in the study, and I think it says a lot that whenever we receive an email saying we're doing this thing that's part of the study, we wondered if Freddie would like to be involved, he always says yes. And being Freddie, he wouldn't say yes if he didn't think it was going to be fun. He enjoys doing it, he likes being involved with it, and I think that's a really big part of it, that the study is fun for the children and they want to be involved in it.
In the first part, we explored whether we could identify cognitive profiles within the CALM cohort. But what about brain profiles? The next part of the analysis that we did was led by Roma Schugstate, who's a postdoctoral scientist working in my group. And one of the brain scans that we acquired as part of the CALM study is a T1 weighted image. And a T1 weighted image is specially designed to image the outer 6 millimeters of the brain, sometimes called the grey matter. And we were able to estimate the thickness, curvature, and the depth of the creases in children's grey matter across multiple different brain regions. So what we wanted to do was explore whether we could map the different brain profiles within the CALM cohort. So we introduced these profiles to the same kind of artificial neural network that we had used for the cognitive data. So this time, the network looks a bit different. There's just 2 layers. The top layer of nodes are these dark purple circles, and the lighter ones are the second layer down. And it got us wondering, well, what's the relationship between these 2 types of space, the brain space that we have just created and the cognitive space that I told you before? For example, if you sit in one part of the brain space, does that predict exactly where you'll sit in cognitive space? Or maybe one location in brain space corresponds to multiple locations in cognitive space. In short, if we knew about your brain profile, would that make a good prediction about your cognitive profile? And the way we tested that was using the same holdout cross-validation that I told you about before. So if you imagine we start with 80% of our sample, we use their brain profiles from the T1 weighted scan to create a neural network that describes those brain profiles. We then take the holdout 20%, we take the first child, and we find their best matching unit within the network. We then use the children who, in the 80% of the sample, created that node, and we use their cognitive profile to make a prediction about this child's cognitive profile. And we then compare it to the child's actual cognitive profile and see how close we are, relative to if we'd just chosen a node at random from the network. We then take the next child, we find their best matching node in the network based on their brain profile. We use the children who created that profile within the original 80%, and we use their cognitive scores to make a prediction about this child's cognitive scores. And we see how accurate we are relative to if we'd just chosen a node by chance. And we repeat that process until everybody has appeared in the holdout 20%. And at the end, we've created two distributions. One is a chance distribution, and the other is how accurate are we at predicting someone's cognitive profile from their brain profile. And this time, the effect size is much smaller, just a Cohen's d of 0.15. That tells us that there is a significant relationship between a child's brain profile and a child's cognitive profile, but the overall size of that effect is relatively small. And that got us wondering, why is it so small? And it made us wonder whether we were thinking about the brain in the right way. So let's imagine other complex systems, maybe a transport network like the London Tube. If you wanted to make a journey one day and say Russell Square was closed, you might not realize unless you were unfortunate enough to need to get to Russell Square. But if King's Cross St Pancras, just up the tube line, was shut, you'd soon realize, because King's Cross St Pancras is a hub station within the network, and so when it goes down, it can have a catastrophic effect across the whole network. And it turns out that these hubs play really crucial roles in all sorts of complex systems like transport networks, telecommunications networks, and even social networks. And it turns out that the same is true of the brain. So we used a different type of brain scan that we had also acquired on the CALM cohort. And this type of brain scan is called a diffusion tensor imaging scan, or a diffusion weighted imaging scan. And if you imagine you had a pipe, water would diffuse down the pipe very nicely indeed, but it wouldn't diffuse easily through the walls of the pipe. So you can infer the presence of the pipe by the direction of the water diffusion. And the same is true, essentially, for the pipes in your brain. So axons become myelinated, they become lined, and they produce pipe-like structures that water will diffuse down. And we can image that diffusion using a DTI, or a diffusion tensor imaging scan, and thus image the presence of those connective fibers. And once we have those, we can use them to create a model of that complex system, a different type of network called a connectome. And we created connectomes for each child in CALM. And within each child's connectome, we could then find the highly connected areas. These are called the hubs. And the areas I'm showing you now are the hubs within the children in the CALM cohort. And one of the really nice properties of these connectomes is that you can describe them in mathematical terms. So for example, we can calculate the overall efficiency of the system. We can calculate how easy it is to get from any one part of the network to any other part of the network, and so on. And so we reasoned that maybe some children's brains are really heavily organized around these hubs, and other children's brains are less organized around these hubs. And we tested that in the following way. So we took each child's connectome, and then we would disconnect the hubs one at a time. And as we disconnected the hubs, we would recalculate the overall efficiency of the network. And what you'd expect is that as the hubs are disconnected, the overall efficiency of the network drops. But it drops differentially according to a child's cognitive profile. And the overall size of that difference is big. So if we return to the original cognitive map that I showed you at the start, children on the far left-hand side of the network, their brains are most affected by the hub knockdown. Their brains are most affected when you remove the hubs, implying that they rely most heavily on the hubs. And as you move from left to right, you find that the impact of the hub knockdown decreases, implying that those children are less reliant on the hubs in their brain. Until you get to the children right at the far right-hand side, you might remember those are the children with the most widespread cognitive difficulties, and those children rely least on their hubs. So there's this general principle to a child's brain network, it seems, that the extent to which it's organized around the presence of hubs seems to predict the overall cognitive performance of the child. So just to summarize that section, we were able to make brain profiles using the grey matter from the T1 scan and show that this can make a significant prediction about a child's cognitive profile, i.e. where they sit in the cognitive network. But that prediction is relatively small in effect size. We can use the diffusion tensor imaging data to create a different kind of network, a connectome, and then explore how this differs across the children in CALM. And in particular, the extent to which a child's brain is critically organized around the brain's hubs seems to predict their overall position within that cognitive network.
So the first time we came was nerve-wracking, because obviously you don't know what you're going to find out. You loved it. Attention, one-to-one, which is what he's good at. And the brain scan, you really enjoyed, didn't you? And we've got the picture at home.
So what we've shown so far is that you can use an artificial neural network to build a cognitive map or a cognitive space that describes the different profiles within CALM. And that the extent to which a child's brain is organized around hubs will predict where children sit within that space. But why? Why are some children's brains more organized around hubs than others? And so far, we've got no theory. And that's what we explored in this final part. And this is work led by a PhD student of mine called Daniel Akarja. And he's been using a different type of network, a generative network.
So if you imagine that we have this diffusion tensor imaging data from each child in CALM, so we can build a really accurate model of their connectome. Could we simulate the growth of a connectome so that it mirrors each child's network within CALM? And that's what we wanted to test. And to do it, we used a really simple wiring equation. So let's imagine that we have two brain areas, I and J. And we can say the probability of those two brain areas wiring together can be expressed as the trade-off between the cost, so the cost of them wiring together, versus the value or the benefit of them wiring together. So for example, in our model, cost is just distance. If nodes are far away from each other, it's more costly for them to wire together than if they are nearby. And the value could be anything, could be any characteristic of a node that might make it useful to form a connection. So for argument's sake, it could be that the node you're wiring to has very similar neighbors to you. So the question is whether we can use a simple wiring equation like this that's computationally specified to simulate the growth of complex networks like we see within the CALM data. So imagine this scenario. Early on in development, node 2 is trying to decide which node to wire to. And because there are no other connections in the network yet, it's simply driven by the cost, so what node is closest. So for argument's sake, node 36 is closest. But later on in the learning process, now that there are a few connections, you can see that as node 2 tries to make a new connection, it could wire to node 25, which is relatively close. But over on the other side, node 59 has a very similar set of neighbors to node 2. So you can see, for example, that node 69 also has many similar neighbors to node 2. So in this case, even though node 59 is far away, the probability of it wiring to it is much higher because of the trade-off between the value or the benefit and the cost, and so a connection is formed. And you might think, well, how can this produce complex networks? And it's because within the model, the addition of just a single connection can radically change the future wiring probabilities. So on here on the left, you can see the wiring probabilities before that connection was made. And on the right-hand side, you can see the wiring probabilities after that connection was made. So a single connection can steer the network down one trajectory or another. I know that's not very intuitive, but if you look at this simulation, you can see the network forming in the simulation. So connections are made between nodes, and the overall size of the nodes correspond to the number of connections that are being formed. So what we can then do is use this simple wiring equation to simulate networks, and then see how similar those networks are to the actual individual brain networks from the children in CALM. And we can compare them using a special energy equation. And if they're very similar, then the energy equation will produce a low energy metric. And if they're very different, it will produce a high energy metric. So we've tried this for multiple different rules, so multiple different values. And what you'll see on the far left-hand side is that one particular type of rule seems to produce the most plausible or the most low energy simulations. And this rule is the one that I described earlier, whether or not the nodes have matching neighborhoods. If they have matching neighborhoods, then a connection between them is highly valuable. And the trade-off between that value and the cost seems to predict very nicely the kind of networks that you actually observe within the CALM cohort. So if we take that single rule and zoom in, you can see the two parameters that govern the trade-off between that rule down the X and Y axis. And you'll see there's that blue sliver. So that's the space in which the low energy networks can be found. And you'll see that it's actually just a very small portion of the overall parameter space in this kind of energy cleft. And if we zoom in, then those little X's correspond to the location of each child in CALM. So you can see that tiny amounts of variability in the trade-off between those two parts of the equation, the costs and the values that are determined by these two parameters, predicts the variability that you see in brain networks across the children in CALM. In short, the complexity that we observe actually emerges from a really quite simple equation. The next thing to ask is, well, how similar to real networks are these simulated networks? And we can compare them on all sorts of different metrics. Do you get what's called spatial embedding? So if the overall network looks like the real thing, do hubs emerge in the right places, for example? And you can see that if you correlate the number of edges across the nodes between the simulation and the observed networks, then you see that they are very strongly correlated. In short, even though we haven't told the simulation where hubs need to be, it learns where to put them just by the playing out of that simple wiring equation over developmental time. So where does the variability come from? So why do some kids sit in different places to others? Now, because the model is fully computationally specified, we can then pull apart the equation and explore why this is. So we can separate out the costs for each child. We can separate out the different values emerging over time for each child, and we can separate out the wiring probabilities. And then we can ask the question, where does the variability come from within the CALM cohort? And we can express that using a metric called the coefficient of variation. So on this graph, you'll see on the left-hand side that there's very little variation in the costs. And that's because, by and large, the brain sizes are broadly similar and the distances between the nodes are pretty much fixed. You'll see that there's much more variability in that value updating, the updating of what nodes become valuable over time. And that when you multiply these together to produce the probability of things wiring together, you'll see this produces very variable outcomes. And why is that? Well, it comes back to that idea that I showed you before. The addition of a single connection can really radically change the future wiring probabilities and thus steer the network down a different path. Now, because we've explored the different brain areas that represent costs and those that represent values, we can then leverage other different data types. So, for example, What we've used here are gene expression data from the Allen Atlas collected in the USA. And what we can explore is whether there are groups of genes whose spatial expression within the brain overlays neatly with the cost profiles of the different brain regions. And we can do the same for the values. So on the left, you'll see for the costs, and on the right for the values. And then we can take those genes and ask, what do they do? Are they just randomly scattered through the genome, or are they grouped around particular cellular and biological processes? And it turns out that the costs are strongly grouped around metabolically costly processes, like ribosome functions and RNA splicing. By contrast, the values are tightly clustered around synaptic regulation. So those genes are heavily involved in the regulation of the formation of synapses. So just to summarize that section, what we've been able to show is that you can simulate a child's connectome by growing an artificial network called a generative network. At the heart of it is a really simple wiring equation, which just trades off costs and values against each other. Space, the parameter space that describes the CALM cohort, is really tight. So a tiny slither of space, variability within which describes the right parameters for producing each child's brain within CALM. We can then explore the different locations of the hubs and show that they actually emerge in the right locations, even though that wasn't something that we specified in the model. And finally, we can then use that information to explore the very many genes that seem to be involved in regulating the different parameters within the equation, the costs and the benefits. So what I've shown you is how we can use different types of network to model the diversity within the CALM cohort. We used artificial neural networks to create a cognitive space that characterizes the different profiles we observe and then link them to things like literacy and numeracy. We then built a different type of network, connectomes, that use diffusion imaging to explore how different brain areas are connected and how they're critically organized around hubs. And finally, we use generative networks to explore how you can simulate the growth of a brain and how complexity and diversity can emerge from quite simple principles that play out in a dynamic way over developmental time. I'd like to thank everybody who has made the study possible. Thank you to the funders, the Medical Research Council, who funded the study, and the other funders who support my lab, the James S. McDonald Foundation, the Templeton World Charitable Foundation, Bailey Thomas Foundation, Gates Foundation, and Cambridge Trusts. Thank you to Sue Gathercole, whose leadership started the CALM study, and the other scientists who've contributed resources to it over the years. And thank you to the very many research assistants, graduate students, and postdoctoral scientists who have helped collect the data that I've been showing today. And a massive thank you to Joni Holmes, who's the head of CALM and runs the show here in Cambridge. And before we finish, I'd really like to thank our participants and their families, without whom none of this would have been possible. And the final word goes to them.
For Freddie, having his needs recognised has made a huge difference in his ability to learn.
He's improved in everything, his behaviour. I think understanding what's wrong has helped us massively.
Eva's always been really incredible at getting through situations, haven't you? She's really clever at getting around things.
If I had a choice between online school and like going to actual school, I would obviously choose online school because it's just so much easier. Like the teachers can help you, but you can still be with your family and be around them when you're actually working.
In order to be able to focus on it and concentrate on it, they've got to be able to find it engaging in some way.
I think anyone who made friends with me now, like a couple of my new secondary school friends, I think had I not told them, in fact some of them I don't think I have, they would think I was a completely normal person. They wouldn't even know that I had ADHD.
It helps me if the lessons are interesting. So if they're interesting, it makes me want to be in the lesson with everybody else.
I'm getting back on track, and I know that it can only get better now.
thank you to everyone that's like done this for me and helped me learn and do what I have to do in school.
But coming here, we realised the differences academically, massively, and what were his strengths and weaknesses, which helped him understand as well as us as parents.
Calm's helped me understand how good I can actually do at school.
My name is Astle Salvesen, and I'm proud to be a supporter both of the Salvesen Mind Room Centre and the Salvesen Mind Room Research Centre. These are two organisations in Edinburgh, the Salvesen Mind Room Research Centre being part of the Edinburgh University. And we have been cooperating and collaborating for a number of years. This evening, it's my pleasure to say a great thank you to Duncan Astle for all the most absolutely marvellous lecture that he has been just given to us. It is most interesting, and I'm afraid to say some of it, you won't be surprised to say, here is above my head. But on the other hand, it makes absolutely such sense. There's no doubt at all that the whole subject that we've been talking about is not just in little boxes, as he has been pointing out, but it's a very wide sphere with lots of individuals having similar but somewhat different experiences while they're learning. This is actually something that Sophie Dow, who is the founder of the Salvesen Mind Room, it was just Mind Room, has been talking about since she thought about the idea in 1998, and then worked through it to 2001 when she actually formed Mind Room. And since that, she went on to organize a conference in 2003 to get international recognition of the subject, or perhaps not recognition, because it had been well known, but people had not been perhaps working together as closely as they should. The angle that we have been working on is in parallel, I would suggest, to what Duncan has been talking about. And it is great that all these cooperations come together so that we can actually find out in the end how these complicated things are tied in together. This CALM study, and what a lovely word that is, just tells it all. It shows, put it again in Sophie's mind, it shows that it takes all kinds of mind to actually interlink, and therefore people learn in a different way. It gets rid of the foibles and the thought processes, the fears, and then people can go on to learn. Duncan, it is marvelous what you have put together and the way you've brought in your team of young parents and actual youngsters who have had the problems outlined has been most inspiring and interesting. The importance of working between research, which you and Sue Fletcher-Watson at the Salvesen Mind Room Research Centre have been doing is one side of it. But then you've got the Salvesen Mind Room itself, which is actually going out into the field, and we have a lot of experience working with the children and trying to work out how they can be helped. It's linking these two things together which makes everything happen, and soon, I hope, we will be solving the problems which are far too acute at the moment. It's said that in excess of 20% of children have these learning difficulties, and we all, in cooperation, want to eliminate them. And so globally thinking about these things and then working together, I'm sure this will be achieved. We, from our point, are looking forward to the thought of perhaps organising another conference, just as Sophie did in 2003, to have another conference in 2023, where this can be explored even fuller. And I hope that Duncan and his team will be involved with that. So we thank Duncan. I'd like to thank University of Edinburgh, and long may we move forward to solve the work that we've set out to do. Thank you very much. We started off last year recognizing that people couldn't clap because some of the audience who we'd be looking forward to could not hear that. And therefore, we just wave our hands to say thank you and well done. Thank you very much.